All right, so the first topic is that what is a virus? And this is the textbook. And again, remember that I put the textbook as recommended. This is a very interesting book in the sense that Principles of Virology. This was the story of 2008-9. And there was really not a real textbook for the virology. Either they were too little, too simple, or they were too much. And in the American Society of Virology meeting around 2008-9, there was an education group. They started doing that. And then Jane Flint, Vincent Recaniello, and others, they came together and just wrote the book, which is relevant for a 400, 600 level course. So that was the birth of that. And now we are at the fifth edition of the book. And the previous edition is also available. You can get it fairly cheap. This will be varies from around $144 to I have seen $164 in between, something in between that. You do not have to buy the book. It is recommended. On the other hand, you can get a cheap used previous edition, which is a 2015. This is 2020. Unfortunately, it went to the print shop before the SARS-CoV-2, so there is no information that we learned about that. But this is a 2020 edition. The previous edition, which is the 2015 edition, that will perfectly work, and you can get it fairly cheap. And somebody when I was doing in the last fall, they said, Dr. Q, I found a PDF copy of that book for $5. I was like, I didn't hear that. I did not have that conversation. There is a copyright statement in there, and these are some of the websites there. You download that copy and then distribute to the whole class for $5, and I don't know what you're doing. So it is up to you. As long as I don't hear it, I don't see it, you're fine. So that's what it is. So indirectly, I told you to do that. Don't quote me on that. I will get fired. So those are illegal copies, absolutely. I mean, like, these are basically scanned copies of those, and I will highly avoid to do those. All right, so that's the primary book, and then this is the field's virology. No way you're just going to go that. This costs $600 for two. These are basically the encyclopedia of virology, and there will be some of the material that I will pull from here. They will be all given at the bottom of the PowerPoint. And if you just see that, don't access your PowerPoint from your Canvas directly. You can just take a look at that, download them, and you will see the notes area. There will be notes. And that's the reason I say that you can get by. You will get a more complete knowledge if you have the book, but you can get by the material that you will need from that notes area. In the introductory chapter, that notes area is very, very less. Some of them even don't have any. But as I said in my last announcement, don't judge the course on that, because you are going to see like 3 pages of note under some of the slides. So it's extensive in there. And I just had to do that because a lot of people actually were struggling, financial difficulty, and I just don't want everybody, if you just can, with an used book, you're most welcome to. But I just didn't want to press on buying a $160 book, and that's why I said it's recommended, not a requirement. All right, so let's get into the real thing. And the funny part is virus infect all living things. And we regularly breathe billions of virus particles. And these are some of the things that I do not have to even tell you. You all know that. And we carry viral genomes as a part of our own genetic material. And there is a very interesting fact for that. So if you have taken cell biology or molecular biology, that you know that in your every cell you have your nucleus. Inside that you have the DNA, and it codes for the genes that make proteins. So how many percentage of the DNA actually contain information that makes usable proteins or the genes which are functional? How many percentage? Anyone? Even close ballpark? Just throw me some number. Anyone? No, I mean, like, nobody is judging you. Just throw me a random number. it is not that I'm just asking you for that you know it. I'm just saying that in a perception-wise, what will you think? We know that there is lots of junk genes, and there are the ones which make proteins in the thing. Anyone? No numbers? 5. Okay, all right, very close. That's excellent. I mean, like, if you said 50%, I will just scratch a little bit. It is about 1.5 to 2%. So think about that. 98% of your gene is considered for a long time as junk. Now we are understanding that they may not be as junk, but they directly do not do anything or translate into proteins out of that 1.2% of genes. And this will be kind of a shocking if I tell that you are more retrovirus than a human being. 42% of your genome, and these numbers are not needed. This is just a fact and just have a conceptual idea that we carry viral genomes as a part of our genetic material. 42% of your genome are the remnants of retroviral element. So what does that mean? So over the period of time, as we evolved, many different retroviruses infected us. So retroviruses, what are they? They are RNA virus, plus-stranded RNA virus. Again, don't worry about that. We'll learn what is plus, minus, all those things. But they are RNA viruses. They go into your body. They change their genome with the reverse transcriptase enzyme that they carry in the virus particle to a DNA. That DNA gets integrated into your DNA for the rest of your life. Classic example, anyone? One retrovirus? Exactly. So once you are positive for the HIV, you will be positive for the HIV. There is no going back. But with all the different advanced therapies, your production of the virus can be suppressed down to a level that where you may not have symptom, you may have a long life with all these therapies, but that HIV sequence is integrated in your genome all the time. And sometimes what happens, that those genome sequences can excise them out, just specifically cut them out, and get inserted somewhere else. So these sequences replicate, basically. And some of them are called the transposable elements, which cut them out and paste in somewhere else. So this is how we acquired these, that there was just only not HIV, there are many other different viruses as well, like say for example, HTLV, human T-cell lymphotrophic virus is another retrovirus. Many other retroviruses infect us. The HIV one is probably the most famous. If you have a cat, then you know that FELV, feline leukemia virus. So suppose you just adopted a cat, and if you just take it to the vet, and he will tell you to do a FELV test. If you have an FELV test, and if your cat actually is positive, there is no reason to give it a vaccine because it's already positive. So it's just a little bit of matter of time. So it may not have the symptoms, or it may have the symptoms pretty quickly. That's the same thing. So feline leukemia virus is another animal virus. It doesn't infect us, but that's what it is. So you can just imagine that your own gene is about 1.5 to 2%, whereas 42% of your genome is retroviral element. And the thing is that does not mean that many retroviruses actually infected human beings over the period of time. Remember that many of them replicate, and that's how the genome builds up and become much more. And the interesting part is that we're going to talk about that in somewhere else in the material. We now know that some of these retroviral elements control gene expression. So they are really not junk. They are controlling the gene expression of that 1.5% gene. And the funny part is that this is kind of mind-boggling, that after the human egg is fertilized by the sperm, you get a 4 to 8 cell stage before it goes into morula, then blastula, then gastrula. In the morula stage, when you have an 8 to 64 cell stage of the embryo, you actually literally see tiny virus particles inside those cells. And we are just trying to understand what they are doing. And after so tiny virus particles in these cells, 8 to 64 cell stage. These are called ERVs, many of them KERVs, ERVs. ERV stands for endogenous retrovirus. Those are in your genome and they express and do something. What the scientists now speculate that at that early stage, when you have an 8-cell stage, 64-cell stage, those tiny virus particles appear and it actually builds the cellular immunity at that level. So think about that we think about that a child when he was born gets the antibodies from the mother. It takes a little bit time for the overall immune system to develop, but the cell is getting primed with its antiviral properties due to the production of those virus particles at the 8 to 64 cell stage. And then they disappear. After that, you don't see them in the blastula, gastrula stage and as the embryo is developing. So these are some of the amazing facts about the viruses. All right, so here is some numbers and a very self-explanatory slide. I do not have to go into the detail of that. But think about that. 10 to the power 30, that number is, I cannot just wrap my head around with that. And it's about a femtogram, 10 to the power minus 15 grams. And the last line, see that. The length of head to tail of 10 to the power 30 phages will become 100 million light years. And think about that. These are in the range of nanometers. So a nanometer size particle, if it is 10 to the power 30, that can stretch that long. So these are some of the mind-boggling ideas about that. So the bottom picture is a typical phage, which is a bacterial virus. And the top picture is a cartoon of a bacterial cell in the blue. And the red tiny things are some of the bacteriophages. There are many, many different of them. All of them don't look the same. But they are infect bacteria. So not only the viruses infect the human beings, all living things, every living things on the planet is infected by the viruses. So you have plant viruses, bacterial viruses, protozoal viruses, everything. So these are some of the other ideas. So you can see that more viruses in a liter of coastal seawater than people on Earth. I mean, like, just to see that some of these numbers, and it's sometimes it's just really incredible. So you can just see the abundance of the prokaryotes, which are the bacteria, and the protists are basically the single-celled protozoa. And if you compare it with the viruses, you can just well imagine that how it is. So the abundance versus the biomass, it's a very, very interesting thing that you can see. But they're tiny, but their numbers are mind-boggling. All right. So here, this is a new word that gained popularity in the last decade, the last 10 years. What is a virome? Now, everybody can tell that what is a microbiome. You already know that in the human gut inside, we have a very healthy bacterial colonization that helps in a lot of positive way, and that's the microbiome, and mainly bacteria. There are some other fungus as well, we are learning. But the virome, we really did not have an understanding that what are the viruses. There are plenty of viruses in our gut, and what they are doing, we are just starting to learn those. And if we just sample a human being just randomly, you can just see that where you get those from. Say, for example, in the skin, hair, nails, you mainly get some of the DNA viruses. And remember that this is a sampling from a perfectly healthy individual without any viral disease. That's the important thing. So all of us in this room, if we just sample, it will be somewhere close. And these are passive agents not doing anything and causing a disease. So this is the interesting part. And another thing that what is important that you can see, that DNA viruses are more compared to the RNA viruses. Remember that this is the first time that we will go into the very detail, and that will be a large part of the course, that the genetic material is RNA for many of the viruses, and that is very different from any other living things, which all have the DNA genomes. I'm going to talk about that. So on the right-hand side is like a very cool experiment that I'm going to show the video in a second. So this is actually a research of the coast of Australia. What they're trying to do, that they have a vessel, and from there, they are looking at some of those humpback whales. And you can see that as they're breathing those spray of these waters, those are coming out. And what they're trying to see, that they are trying to see that in the whale breath, in their respiratory system, what kind of viruses are there. So they're studying the virome in these humpback whales. So what they do, that they actually send a drone where they have a lid which they can control, and they are sending that through that mist of the whale breath to collect the viral samples. And it's really pretty cool. This is how the research and the sampling are done. So you can just easily see that. So you can just easily see that it just, again, I will just play it one more time. So this is how you collect the sample, the drone comes back, you get those viruses and analyze them in your laboratory. All right, so this is the fact that I was saying, that when we are just sampling, the vast majority of the viruses, in fact, have little or literally no impact on our health and well-being. And why not? The reason for that we are going to elaborate, that they are passive agents, means that they really do not do. There are lots of chemical reactions need to go on, and all of them are chance factors before a virus will infect you. So they are very, very specific. So that's the reason that we really do not have any impact on that. So what you are going to see, that this is a cartoon from, all these viruses are cartoons from the principles of virology, and you are going to see these cartoons all throughout your course, and they are drawn into pretty much based on the structure of the viruses. So they are just not just plain simple contour, that it's not an artist rendition. Many of these viruses structures are solved by the cryo-electron microscopy at the molecular level, and they are based on that. So let's see somewhere here. Can you identify the coronavirus in the picture? Which one is the coronavirus? Up, down, top? Okay, I'll explain that. How many people saying it's the top? And how many people are saying it's the down? And which one is in the middle or the top? Okay, all right, let's not do shenanigans. Let's do that thing. So it is actually at the bottom. So this is the coronavirus. So, and I will just explain that why this is not. I will just give it and explain. It's very difficult until you have gone through the very detail of that. They are looking very, very similar. So this is the coronavirus. The sticking out is a trimer. That's the spike protein, which is a trimer, three of proteins together. And then you have an envelope protein as well as a matrix membrane protein. And you have an envelope outside, and this is derived as the virus buds out of the cell. This is stolen phospholipid bilayer as it comes out of the cell. And inside that, you have in between a 28 to 34 kilobase genome. Very, very large genome. The largest genome of all RNA viruses are the coronaviruses, and it's extremely complex in that sense. So that's what it is. And the advantage of that, now you can understand that why we say that just hand washing soap water or alcohol is going to take care of any surfaces which are having the coronaviruses. The reason for that, it is fairly easy to destroy envelope virus. So here, just below the protein layer that you have, all these proteins are embedded into this membrane right here. And this membrane is phospholipid. And all you know, soap, detergent, they take away grease. So basically, these grease or the soap, they are amphipathic molecules. They have a polar head group and some hydrophobic tails. They just get inserted into the membrane, alcohol as well, hydrogen peroxide, any of the disinfectant, they're going to completely disintegrate the membrane. As soon as that membrane is disintegrated, it loses its infectivity completely and it's destroyed. So destroying enveloped virus is far more easier. Whereas there are just a couple of pictures, say for an example, if you just think about adenovirus or poliovirus, these viruses are the naked viruses, doesn't have a lipid layer on the outside. These are made up of protein shell capsids, completely protein shells. These are very resistant like compared to the enveloped virus, more resistant to chemical treatment. Just alcohol spray or just soap will not destroy them that easily. You need a much more drastic effect. Hydrogen peroxide will do, but that's what it is. Some of the interesting stuff, on the other hand, why this is not. So this is actually a herpes virus. So what is the difference in between that? The herpes virus is a DNA virus, and what you see that inside you have a hexagon, and this is basically a capsid made up of a protein shell which contains a double-stranded DNA. Then you have a tegument surrounding that, and then you have a membrane, and then you have the membrane proteins in there, which are known as the H1, H2, H3s, many of these proteins. So this is a DNA virus, can be very, very large, and the genome size could be more than 100 kilobases, way larger. And remember, your chickenpox virus, varicella zoster virus, is also a herpes virus. So that's it. So right here, this is an HIV with a dual genome. And again, you do not have to know that. This is a variola. This is your pox virus, which causes smallpox type of thing. Right here, this is the influenza, which is also an RNA virus with a negative strand, and there are 8 segments of RNA in there. See that these, the coronavirus have a long 30 kilobase approximately genome, whereas these are segmented. So many different types. You will see that some of the genomes are incomplete. You have a DNA genome, and a part of the genome is DNA. You have a green, that part is an RNA. So you can see that they come in all shapes and sizes with these. And we're going to talk about many of these as we go on. So as I said, that most viruses actually pass through us, and they are all passive agents. And we ingest of many of these non-animal viruses regularly with food. And the greatest amount of material or the virus that we ingest comes from the salad or the raw food, fruits, vegetables on their surfaces. Even after washing, there's plenty of viruses. They don't do anything. So for an example, right here, the most common ones is that is a baculovirus. And this baculovirus is a double-stranded DNA virus, very long. And it infects those cabbage loopers. And they are found all over the cabbage. And this is one of the greatest source, like say, for example, if you're just eating coleslaw or any raw cabbage, any kind of cabbage, many of them, and you are going to get those baculovirus. But this infects cabbage loopers. They are insect viruses. It doesn't have any protein, and it doesn't do anything to the human beings. So they just pass through us. So on the other hand, you can see that the metagenomic analysis, basically you just take the samples from a stool and you just run and just do a deep sequencing to find every possible viral sequences in there. You're going to see that most of these RNA viruses that we find are similar to plant viruses. That's very interesting as such. So you can just easily see that 91% resemble plant viruses that from just a single study. And you can see that the most abundant human fecal virus is a pepper mild mottle virus. And you know that it comes from the black pepper. It is like so ground, even the black pepper, when you're just spraying that, you're just ingesting that in a large amount, doesn't do anything. But these are some of the funny facts about the viruses. They are all passive material. So we are saying the passive material. I mean, like we started that, okay, all right. Whenever we talk about the viruses, that they are always bad. That is the common thing. So we already saw that there are passive agents not doing anything, not doing any harm, that like I said, plant viruses, insect viruses, not going to do any harm to the animals or the human beings. But are there any good viruses? And that's the interesting part, that we already talked about that. These ERVs, or the endogenous retroviral element that we have in our genome, they control the gene expression. But again, remember that they are not an intact retrovirus. They have lost some of their genome. Remember that. If you have an intact retrovirus, you are going to produce a retrovirus as such. But all these 42% of the genome that we talked about are the retroviral elements. Those are remnants or defective copies of the retroviruses. If you do not have a defective copy, you are going to produce the viruses as an adult as well. Say, for an example, an HIV-infected person will have that sequence in there. But what will happen that over the period of time, these sequences basically get lost, and due to the mutation over the many years, and lose the ability to produce any viruses, and they are all defective. On the other hand, here, a couple of examples, and we are just starting to learn these things, that are there any good viruses? So here is an example of a three-way kind of symbiotic relationship with the viruses. So this is actually a very tall grass, Dicanthelium lanugism, and they grow in the geothermal area of the United States, say Yellowstone National Park, where they're close to these hot water geysers and the hot water mud, those springs and all those kind of things. And the soil temperature could be unusually high, like 55 degrees Celsius at that time, very, very hot. So what happens that there is an interesting thing, that this grass is colonized by a fungus, which is Carvularia protuberata. So the name of the grass is Dicanthelium, and Carvularia protuberata is a fungus which basically has a symbiotic relationship. So basically the fungus lives on the grass, gets its nutrient from the grass. And then the third agent is right here, a virus, which is a Carvularia thermal tolerance virus. And this virus actually infects the Carvularia. And when this virus infects Carvularia, this gives the thermal tolerance to the grass. And so it's kind of amazing in the research that they came up. So basically, if you try to grow the grass in the laboratory in that similar condition, you have a Dicanthelium and you have the Carvularia. Without any viruses, they will not grow. You need the virus infecting the Carvularia so that they can colonize and grow. And these are long grasses. They could be up to 3 meters tall, like 8-9 feet tall grasses. So this is one of the interesting part, that where we see that there is some symbiotic relationship with the viruses. There is also another interesting part. This is from a Nature article, and this is the first time that we just understanding that, at least from the germ-free mice, there are some ideas about that a norovirus is doing something with their gut. So the norovirus is basically, we call this a cruise ship virus. So sometimes in the cruise ship, these gastrointestinal disorder break out. There are many other viruses that can cause that. Rotaviruses can cause that as well. But norovirus is one of the common one that happens in the cruise ship as well. So here you have a murine model, that means a mouse model, and let's see that what those are. So on the right-hand panel, what you are seeing, that these are stained by the hematoxylin and the eosin. So that's a standard histological stain. And a healthy mouse right here, so see what you are seeing, that this is a cross-section of the intestine, and you are looking at these finger-like projections, which are called the villi or the villi. Now, each of these villis, finger-like projection, is lined by hundreds and thousands of individual epithelial cells. And each of these epithelial cells, which you are seeing right here. So if you look at this right here, the pink part is the cytoplasm, and the dark bluish part is the nucleus that you are seeing. And on top of that, that where I'm just putting the cursor or the arrow is what we call as a brush border. All these epithelial cells do have microvilli, like hundreds of microvilli. So villi is the largest structure, much larger finger-like projections, not as that big, but those are visible under just a low magnification of microscope. Increase the surface area of the absorption, and then you have the microvilli on individual epithelial cells that basically absorbs the food material and the digested food material, and it increases the absorptive surface almost 600 times. So you can see that right here, this is a very healthy condition. So all these cells look plump and nice. The brush border looks nice. Now here is a germ-free mice where you don't have any kind of bacterial flora in the gut of that. This is, you can grow them in the laboratory condition only. No, there is no bacteria, no virus, there is no microbiome inside their gut. And you can see that how sickly these cells are. They are you have some of these gaps in between them. They look much sickly compared to that. And you can see that the thickness is much lower compared to the thickness. So basically the absorptive performance of this will go down. But now what happened that if you just infect these germ-free mice with the MNV, which is the murine norovirus, it doesn't infect human beings. This is for the mouse norovirus. And you can easily see that they actually get better. So you can compare to this picture from the germ-free without any norovirus. And when you add the virus, and of course this is the control contains the norovirus. But here, from the germ-free, no norovirus to norovirus, you can revive them back to almost normal cells. So this is doing something to these. And these are the things that the gene expression of many of these structures may depend upon a viral infection. So this is a first example of that gradually. So the bottom two panel, what you are seeing, that the CD3 is a marker for the T cell. So these are stained by an anti-CD3 antibody conjugated to a dye, which is this dark brown in color. So in the conventional cell, you can see that the central part of the villi contains the T cells, and these T cells are basically the cytotoxic T cells, as well as some of the helper T cells. They basically are there to their surveillance cells. So basically, if it get infected, it's going to alert the immune system to do something. So you can see that there are plenty of T cells in a healthy structure, and in the germ-free mice, basically there is none. You do not see those dark spots which are stained with the anti-CD3, which labels the T cells, very less. But when you basically infect with the norovirus, you can see a healthy number of T cells return into that area. And this is what I was telling, that very early at the cellular immunity, that your endogenous retrovirus at the 8 to 64 cell stage when expressed, your cellular immunity at that level, you don't have T cells yet. They have not differentiated into that. But these gene expressions of some of the cells get programmed for antiviral defense mechanism that early into an embryo. So this is also another beneficial function of the commensal bacteria. So this bacteria is basically doing something. The bacteria does do something, but the norovirus actually just do the same thing, just like the commensal bacteria in the gut. All right, so some of the interesting part that the viruses are amazing molecular nano machine, but I mean, like at this point with this pandemic, we just like, if we praise the virus, it just becomes very hard for the people who are hard hit by the pandemic, and that's definitely true. And what you're seeing that when you're looking at this picture, this is just not artist rendition. This is basically a molecular model. So what you are seeing right here is a virion, and these are from the cryo-electron microscopy. So each individual colored parts are individual protein in the virion. That's what you are seeing. So you can see that there's a five-fold symmetry right here, turret-like structures. And a virion is, again, by definition, is an infectious virus particle. So we are going to use that. An infectious virus particle is a virion. So that's what we are going to, I'm going to talk about. So if we just talk about just the virus, it could be, say, in a test tube, we say that there are 10 to the power 6 virus particles. That means that those 10 to the power 6 virus particles, they could be infectious or non-infectious. But if we say that their virus 10 to the power 6 is the virus and 10 to the power 4 are the virions, that 10 to the power 4 virions technically are intact and capable of infection. And these numbers are much less than the total number of viruses. So say for an example, most of the viruses that basically just we go out from our body or any reservoir, most of them are defective. But the sheer number that if you're just spreading 1 million virus, you just need a couple of viruses to be effective in that. And these numbers also vary. We're going to talk about that. So some of the viruses inside an outer shell also do have an inner shell. This is a reovirus, and we're going to talk about that how it is. And why it is an integrative science, because when you are solving the structure of the viruses, the earlier people tried to do X-ray crystallography, now is cryo-electron microscopy. You need engineering basically to build these machines, and then you need physics background, chemistry background, and lots of computation to decipher these structures. So these are the structural biologists which are very integrative. On the other hand, there are many other things. Say, for an example, we have the surveillance from the sewer water for the COVID for the COVID-19 or the SARS-CoV-2 in our sewer. Just gives an idea. That strategy was developed in the Department of Environmental Sciences, not biological sciences or biochemistry. On the other hand, in the other department, chemical engineering, biomedical, then your bioengineering department, they are all doing, even in the computer science, there is one faculty member trying to develop some biosensors on these viruses. So it's a very integrative science. All different disciplines actually work on these, basically. All right, so this is the overall course goal, is designed to help you with the big picture of the virology and how to think about virology as an integrative discipline, not an isolated collection of viruses, disease, or genes. So the classical virology courses, like 20, 30 years back, or even how it was taught that you just parade a virus every day. Okay, all right, today we're going to study bacteriophages or the bacterial viruses, a couple of lectures. We're going to study plant viruses. These are the plant viruses. Then you go to animal viruses. You just, okay, today poliovirus, next day herpes virus, next day pox virus. And we do not study that way anymore. The reason, those are some of the facts that you can look up at your fingertips, like literally. You just need to see pox virus, you just Google that, just go to a wiki, and then it will take you to some of the references. You can just dive deeper. They're available to you all the time. But the course goal here is basically to understand the underlying principles of how these things work. Basically how they infect, what the changes in the cells that they are doing and what they are doing. So we are going to learn that, and that will universally expand your knowledge to any of the viruses that you are going to study in the deep. So that's the difference in that, because most of the viruses follow these set of rules and the principles. That's basically what we are studying in this class. And learning the fundamentals about these molecular wizards that amaze the informed and frighten the uninformed. Sometimes that's what it is. So you're seeing that there's a BSL-4, Biosafety Level 4 laboratory, where you have a complete 2-layer of isolation, and you actually carry your own air. So those air lines are on the ceiling, and you just have an air hose and you breathe in that. So if you're just using SARS-CoV-2 in the laboratory condition, you still don't need, it's not a BSL-3 agent. It is still a BSL-3 laboratory can handle those if you're using a live SARS-CoV-2 virus. But if you just do not have a live virus, even a BSL-2, like if you're just working on the genome or other proteins or something, even a BSL-2 laboratory will allow you to do that. This we are talking about, say, Ebola, Lassa fever, that kind of virus. So there is a couple of BSL-4 laboratories. One, the latest one is in Boston University, and one is in Galveston, just across from the bay. Right there is a BSL-4 facility, highly secured, because many of the agents right here can be used as a bioterrorism agent. So very, very secured. All right. So here, debunking the misconception. And it's kind of funny, and this slide is a controversial slide because people have laughed on that. So I could have picked up SARS-CoV-2 instead of a swine flu. This is from a 2009 slide from CNN. And it would have started a whole set of controversy again. And I just wanted to stay away from that. So you do those things in between yourselves. So here, we are not concerned about this. So this is the H1N1 flu, that's 2009. And see what happens that, okay, ravage lungs, spreads through respiratory system, causes lesions, doesn't stay in the head like seasonal flu. This is not a neurotropic virus. What do you mean by doesn't stay in the head like a seasonal flu? Your seasonal flu influenza goes into your brain? No, it does not. So these are some of the ways that when media poses that, you get that and you talk to people, and this is how even a credible source will do that. Now, when I said a credible source, people say it's like, so there are two things. Either you hate CNN and watch Fox, that could be me, or you watch CNN, that's why you picked it up. I'm like, you'll guess. I'm not going into the controversy in that. So here the question is that whether I am pro-CNN or anti-CNN, and that will start a whole new ball game of that. This is just a internet picture, not even taken by me. This is from a slide from Vincent Recaniello, who is one of the author of that book, and he's a Higgins Professor of Microbiology, Immunology in Columbia University in New York. And this course is built on the basis of his course and book as well. So this is from, I stole this from there. Right. So the definition of virus, we're going into some real terms. So infectious, obligate, intracellular parasite comprising of genetic material DNA, RNA, surrounded by a protein coat, sometimes a membrane. And the first thing that you are going to see that the viruses, as I said, that they're passive agents. It's a molecule. It does not have any life. Then how can you say that it's a parasite? That's a term that we use for a living thing. Unfortunately, we just really do not have a word. Sometimes you can say things like, okay, it just competes with the cellular machinery. And that's, as soon as you say compete, that's again just gives them a word that you use for a living thing. So it's very difficult to sometimes define. But these are some of the definitions. You can say that to refine it to some extent. An infectious, obligate, intracellular, molecular parasite, that's it's there in another slide. Molecular parasite. The molecular parasite means we are defining that it's basically a collection of molecules, which are the proteins, lipids, and the carbohydrates, and not a living entity as a parasite. So that's the best definition that we can do. And you can easily see that we already talked about that coronavirus, that there are some of the naked viruses like these. These are surrounded by a protein coat right here as well. These are non-enveloped right here, this one as well. These are non-enveloped. They do just only have the protein coat. They are much more difficult to destroy. On the other hand, the ones which are usually round long have we call them a helical symmetry. These are surrounded by a membrane, and this membrane is almost exclusively always a phospholipid bilayer and derived from the host. Now remember that derived from the host does not mean that it is always derived from the cell membrane. This can be derived from the endoplasmic reticulum, this can be derived from the Golgi, or any of the endomembrane system as well as cell membrane. So this can be derived from anywhere, but it is usually always exclusively the endomembrane system of the host cell. And then what happens that very interestingly, as the virus matures, so we're going to talk about that, all these proteins that is decorated outside, particularly the enveloped virus, they do not, the virus don't exit the cell that way. These proteins which are on the surface usually stay inside the membrane. So you have a phospholipid bilayer, they stay inside. And there are some of the enzymes called the flippase, as well as there are some of the areas which we call as a lipid rafts, which actually after the virus matures, these basically flip or move through the membrane to the outside of the surface. So for example, just giving that how it is, that as an HIV is budding out of a cell, immediately after it buds out, if you take an HIV, it still doesn't have those proteins. These proteins are not there yet. They are still inside. And at that time, the virus will not be infectious. The same way for the coronavirus. As it buds out of the cell, immediately it's not infectious. These spike proteins are inside. And with a short period of time, and this short period means just like, not hours, just like a few minutes, they will be flipped towards the outside. And once they are, we will call them a virion, which is the intact viral particle. Sometimes we call is a mature virion. Sometimes there's a mature. So this process is called a maturation of the virus as well. All right, so here is the definition. Once again, we just try to add that molecular parasite. But again, obligate means that there is no other place the virus can replicate. Means that if you just have a body of like a tissue fluid, or even if you have a blood, the plasma, which doesn't have any cell, if you put a virus in there, it cannot do anything in there. You have to have a cellular machinery before it can replicate. This is not the case for a living animal. If you have a fungus or you have a bacteria, if you just have a rich plasma, you just put some bacteria, it will grow in there. Even in some media, it will grow in there. But in some bacterial media, if you put a virus, and if there is no living cell in there, it's not going to replicate or do anything. So that's why it is an obligate intracellular parasite. It has to go into the cell to perform its functions. So this is the virion, is the infectious virus particle, and the reason that why the virus is not an organism is again because it cannot live on its own. If it's sitting on a surface, it is just like a grain of a sand or a grain of a sugar or a grain of a salt. If that touches completely by accident, completely mechanical, it doesn't have any brain, a cell or a surface where there is a cognate receptor where it can bind, and this is completely by mechanical movement, either air current or touching and all those kind of things, mechanical movement. If it touches the receptor, at that time, the cell will take the virus inside. And that's the process. The binding takes the virus inside, and once it is inside the cell, it will do something. So here we are going to see that these pictures a lot of time. You can see that caveolin-based specific type, you have a receptor binding, and the virus is entering, and it takes these microtubules to travel into an endosome. From there, the genome is released into the nucleus. The genome is replicated. On the other hand, it makes the mRNA. The mRNA makes the proteins. These genomes also make some of the late mRNAs, some early mRNAs, late mRNAs make more proteins, and then the virus is assembled and just getting out of the virus. So they are taking all these molecular machines of the cell is completely hijacked. And that's why we say that the cell is basically under the control of the virus. And this is typically the parasites do. They benefit themselves while harming the host. And it will shut down pretty much all the functions of the cell. So basically the ribosomes that you are seeing, these are the ribosomes with the mRNA green. They are basically destined to make only viral proteins and the viral, only the viral proteins. And they will shut down the machinery and the cell will eventually die. Some of the viruses kill the cells very quickly, some of them slowly. So these are some of the ideas. So on the left-hand side, so the right-hand side is a complete cartoon based on real pictures, whereas the left-hand side is actually a structure from the cryo-electron microscopy of a virion. So here, this is one of the important that we avoid this anthropomorphic analysis of the expression, but again, I am just, I already said that we are using the word parasite, or it competes for the host, and we're just using, we cannot avoid, but in most senses, just keep in mind that the viruses do not think, they do not employ, ensure, exhibit, display, but unfortunately, we do not have a term that we exclusively use for a passive agent or a chemical molecule. They do not achieve their goals in a human-centered or an animal-centered manner. They are passive agents, as I keep saying. They are not an intelligent entity. So that, for example, that as the SARS-CoV-2 is going on, the Delta variant is like the virus is so smart in there. We always say that. But well, it just happened accidentally by chance. The virus never thought of making a Delta strain. It just mutated into that and becomes infectious. That's what it is. And a viral infection is a series of chemical reactions that randomly took place to make it happen completely by chance and selected and stayed over by the years of evolution. So that's pretty much the idea about the virus. So just a couple of slides that how small they are. So you can see that right here is a red blood cells. You have an E. coli. Here is an E. coli bacteria, a bacteriophage. That's very, very large virus. This is a smallpox virus. You can see a poliovirus, tobacco mosaic virus, a bacteriophage MS2, and bacteriophage T4, much larger. So see that one of the largest viruses that we used to know for a long time is about 300 nanometer by 200 nanometer smallpox viruses. So you need electron microscopy to do that. So the definition of the viruses, well, all throughout the history is like, okay, all right, they are microscopic. Well, not microscopic, they're submicroscopic. You cannot see them with a light microscope. You need an electron microscope to see them. Well, that definition is going to change, and we are going to see that. This is another picture that on a pinhead, you can easily see that you can just fit 500 million rhinoviruses on a pinhead. This is a dust mite, you can easily see. So here what you are seeing, pollen, red blood cells, yeast cell. These are some of the bacillus bacteria, some coccus bacteria. This right here is an Ebola virus, filamentous bacteria, filamentous virus. That's the only virus that you are seeing, pretty much. So you can easily see that when you sneeze, you fire an aerosol contains enough viruses to infect thousands. That's one of the things that we are experiencing and doesn't need any explanation for that at all. On the other hand, that they are not small as we once thought. Pretty much around 2008, the first idea of these giant viruses came, and after that, people started discovering that. So one was discovered from the permafrost in Siberia inside an amoeba. So these are the viruses for the protist. Then there was another pithovirus that was discovered in different sources, but the interesting one, that one patient had an infected eye and has a swollen eye, went to the eye doctor, and the eye doctor isolated a protozoa from the eye. It's an acanthamoeba. And after isolation, they found that there is some interesting thing in there. And they found another giant virus, which is known as a pithovirus. So here, these are the mimiviruses, and you can see that This is compared to another virus, is a chlorella virus. This is a paramecium, which is a protozoa. This is a protozoal cell, and you can see that. So you have an electron-dense material, that's the genome material. And many of these viruses were discovered in France, and then named those viruses as Mimi viruses, Mumu viruses, Momo viruses is funny names in there, but there are some of them we will going to see. There are picoviruses, Pandora viruses. Those are a little bit different in shapes as well. So here is the Pandora virus, giant virus, and you can see that this gave the definition, changed the definition for viruses forever. So this is a 2 micron. Under the light microscope, you can see this virus. So you really, if you just say that they are submicroscopic, if you say that definition, you have to say that there are exceptions. Some of the giant viruses can be seen with a light microscope. So we're going to see that these are also infect the amoeba. You have an apical pore. We call this as a stargate through which these genome actually comes out basically through that. And they have a tegument-like material. And there is also an evidence that they also do have cellulose, and along with the proteins and the membranes in there, and how they synthesize that cellulose inside an animal cell, that still people are working on. Very interesting ideas about those things. Very, very big giant viruses. So the viruses, so I will actually basically, I have 1 minute left. I will not finish the slide in 1 minute, but I will stop in this one, and we just have a couple more slides, and we will go from there. Do not want to rush, and then we will go from there. Any questions, folks?